As you work through the course, you're going to find that DNA and RNA are split into different lessons. You've got several lessons on DNA, you've got another lesson on RNA, you've got another lesson on nucleotides. And the risk with this is that you don't build a cohesive overview. So what we're going to do today is we're going to take 30 minutes to look at DNA and RNA and nucleotides all in one great big smush. Okay, so we're working at a not so fine level of detail as is in the lessons. You do need to know that level of detail, but we're taking a step back and looking at how they compare. It's one of those wonderful questions that they love to ask, is comparisons between DNA and RNA. So starting off at the very beginning, all of these are built out of nucleotides. So let's have a moment to think about nucleotides. So that's our last biological molecule that we study. Carbohydrates, you've done lipids, you've done proteins, and now you do nucleotides. And we are looking here at a three-part structure. And a lot of this should be familiar. Your phosphate group. Same circle as we drew in a phospholipid. This one I'm hoping is familiar. This is a carbohydrate. We're looking here at a pentose sugar. And this one is our new one. This rectangle is standing in for a nitrogenous base. So a little bit more about each of these. Remember when we draw these ring structures for the sugar, we have that oxygen bridge that's hiding. Okay, so this here is an oxygen bridge. Here we have carbon 1. So 2, 3, 4. Then remember the last carbon sticks out. So hiding here is carbon 5. And it's important that you keep that in mind, because what we talk about is often with reference to the number of a carbon in the position of that sugar. So we don't need to know the exact detail of the structure of the sugar, but we do need to know where these carbons fit in, because that helps us to understand the structure and the bonding within the DNA. So we have beautiful covalent bonds here, of course. So let's get another sort of, we have a star there, and we have a star here. And those are our covalent bonds. So we have a really strong molecule here. This is sort of one unit. We think of it as a nucleotide. Yes, it's made up of a phosphate, a pentose sugar, and a nitrogenous base, but they're bonded together covalently. So we tend to work at the level of this is one unit. A little bit more detail within this unit. Okay, for our pentose sugar, we have two options. We have, in DNA, we have deoxyribose. And in RNA, we have just plain old ribose. What's the difference between the two of those? Deoxy, less oxy. So it's all about carbon 2. Okay, so carbon 2 sits here in this little corner here. So he's got an up and he's got an along. But he's also got, in deoxyribose, 2 hydrogens. Whereas in ribose, carbon 2 has a hydrogen on top and a OH on the bottom. So there's your oxygen that's been lost when you get to the deoxyribose. Phosphate is phosphate. Nitrogenous base, this is where we can also get some more fun and games. Here you have two options. Let's make them fit here. We have purines and we have pyrimidines. Okay, purines, short word, larger molecule. Pyrimidines, long word, smaller molecule. We don't need to get into the details. We do like to know that the purine is a double ring structure, whereas the pyrimidine is just a single ring structure. So you can see there's quite a difference in size. They have the same basic first ring, but then the purines have a second ring. So they're much longer in terms of the distance that they move out away from this pentose sugar. Who's who? We have adenine and guanine are our purines, whereas thymine, uracil, and cytosine are our pyrimidines. And this is stuff that you really have to just sit down and learn. Okay, adenine and guanine sort of make a wah noise and an ah noise and a purine. So that's how I remember those ones go together. Whereas pyrimidine is rather sort of spiky. So it gets the thymine and the cytosine. And uracil and thymine are best buddies. So they have to go together. So that is the structure of our nucleotides. Okay, in order to make DNA and RNA, we have to link these nucleotides together. So let us talk a moment about joining nucleotides. Brilliant for when you get into learning about transcription and DNA replication as well. So if we have a nucleotide, okay, remember we talked about naming by our different carbons. So the ones we want to really pay attention to this time now are carbon 5, and this is now carbon 1, 2, 3 sitting on the bottom. Because those are the ones that we use to name. Now remember, everything is catalyzed by an enzyme. So our enzyme here, for example, is DNA polymerase. We also have RNA polymerase. Lovely one. A is for the enzyme, polymer, what you're making, DNA or RNA. So an enzyme always has a very specific shape for an active site. So it can only join certain things together. That's it, the only way it's going to fit. In this case, it always joins onto a C3. So we would have another molecule sitting underneath, okay, with also its C5 and C3. And our bond that's going to form is going to be here. Okay, so this, we're catalyzing it between C3 and the phosphate group. And it's called a phosphodiester bond. This is another covalent bond. Okay, so what we get is a string of covalent bonds, phosphate to sugar, phosphate to sugar, phosphate to sugar, or phosphodiester bonds. So it's a really strong backbone. For our molecule, or covalent bonding. What we say also, another few bits of words, is that we're doing a 5' to 3' extension. Okay, so we're always building from the 5' to the 3' because we're moving, we're adding on to that 3' all the time. So we're growing sort of down. Okay. That is because our enzymes have a specific shape. So they can only catalyze this reaction in one direction. So we're always building on the one end. So when you do DNA replication, then you get into this in a bit more detail about how the enzymes work and how it does everything. Today we're just looking overview, we're joining our nucleotides. And the take-home message here is that when we join nucleotides together, we have a phosphodiester bond, which is covalent. So our nucleotides in one string are strong. So let's talk about now, we've joined our nucleotides together, we can get either DNA or RNA. So we're going to talk about DNA first. deoxyribonucleic acid, DNA. So DNA is one of those very, very, very few and far between acronyms that you can use, but you do need to know the full spelling as well. If you're asked for it in full, you need to be able to give it in full, correctly spelled. So DNA is a polynucleotide. Its monomer is nucleotides. Put them all together, make a macromolecule, which is a polynucleotide. It's a special one, it's DNA. So when we come to DNA, we actually have several million nucleotides. This is a big molecule. Okay, they're formed into a double helix. So you have two strands, and they are twisted. We have the bases A, T, C, and G, adenine, thymine, cytosine, and guanine. We also have deoxyribose as a pentose sugar. So the thing is, these things are things to remember, because when it comes to RNA, we have a slightly different list to this one. So let's talk about another few funky things when it comes to DNA first. We talk about DNA structure having antiparallel strands. Remember, we just said we have 5' to 3'. Okay, so antiparallel means they're parallel, as in the same thing like parallel lines in maths, they never meet. Anti just means that they're going in the opposite direction. So if this one is 5' to 3', this one will be 5' to 3' in the other direction. I'm not going to try and draw this freehand now, because I know I'll make a stuff up of it, because there's not enough time to do it beautiful. But you've got it in your textbooks, you've got it in your notes. So go have a look at how everything flips around to bond. Okay, so these antiparallel strands are twisted into a double helix. Next thing, there is an exact distance between those two strands. Remember, we're talking parallel. So the distance has to be exact between both strands the whole way. And what's really funky is that there is only a precise enough amount of space for one purine and one pyrimidine. Remember, our purines are the big ones. And our pyrimidines are the little ones. What this means is that, for example, we always have an A or a G. And opposite, we would have to have A to a T, G to a C. It gets even worse than this. Okay, how do we make sure that we've got the right pairing? It's the number of hydrogen bonds. A to a T is 2 hydrogen bonds. G to a C always makes 3 hydrogen bonds. Remember, dashed lines for hydrogen bonds because they're not as strong as covalent bonds. This is what we call complementary base pairs. So we have only enough space for an A and a T or a G and a C. So they can be the other way around. They could be TA and CG. That doesn't matter at all. But if we have an A, the only thing that's going to fit and be able to bond is a T. If we have a G, the only thing that's going to be able to fit and bond is a C. So we have a very specific way of keeping these the exact right distance. And all of this is helping to maintain the code. Okay? Also remember that when you build up a whole lot of base pairs, we've got talking several million nucleotides here, right? Now, if we're talking several million, the minimum number, even if we just had A's and T's, is double that of hydrogen bonds. Maximum would be triple that of hydrogen bonds, because if it's all GC. So we're getting here a lot of hydrogen bonds. And when you have a lot of hydrogen bonds, you have great strength. So that's something to remember is that hydrogen bonds, particularly DNA, is rather like Velcro. So if you pull at the end of one piece of Velcro, it's easy to undo. But if you try to pull the whole piece of Velcro apart at once, you've got a problem on your hands. So something to remember here as well. Hydrogen bonds may be weak in low numbers, but in high numbers, hydrogen bonds are really, really strong. So we have a very specific structure caused by our base pairs, which is strong and stable because of our hydrogen bonds. Also remember, we've got that phosphodiester backbone on either side. So you should be getting the impression here that DNA is a really strong and stable molecule. Crucial. Okay? In prokaryotes, that's the end of the story. We just have a simple helix. In eukaryotes, things get even better. Okay, here our double helix is wound around and further supported by more proteins. Okay, we condense it into chromosomes and we tuck it inside a nucleus. Even more protection. So here we're going to sort of put that hint into your brain, okay? More support, more stabilization, it's going to last longer. So think about the lifespan of a prokaryote versus the lifespan of a eukaryote. The longer you live, the longer your DNA must last, because the DNA is what has all that coding in it for everything you're ever going to do. So stability is important, and the structure of the DNA is just going for it. It's stable, it's trustworthy, it is strong. Okay, so let us then talk a little bit about RNA. So RNA in full is our ribonucleic acid. We're going to start out with the exact same features as we did for DNA. Here we have a polynucleotide. But instead of being several million, we have 100,000 nucleotides. So this is a much smaller molecule. We don't have two strands anymore. We have a single strand. One single backbone. Okay, remember we had ATCG for DNA. Now we've got a UCG. Uracil, not thymine. Also, we have, of course, ribonucleic acid. Our sugar is ribose for RNA. Then what we've got is a bit of fun. Okay? When it comes to RNA, we've got three different types. We've got mRNA, we've got tRNA, and we've got rRNA. So, M for messenger. He takes the message. T for transfer. He does the transfer. And R for ribosomal. He's involved in the ribosome. So, our mRNA, we draw. He is so easy to draw because he's just a line with the bases represented. Okay, so he's the one who takes the message, takes the message from the DNA in the nucleus out into the cytoplasm. tRNA is a little bit more complicated. Still one single strand. Okay, but see here, he has zones where there's hydrogen bonding. So we have a little bit more stability than we do for the mRNA. rRNA, I'm not going to draw him. Okay, he's built into ribosomes. So they will just draw a ribosome for the fun of it. Okay, so it's rRNA and protein that build our ribosome. So mRNA, the only point of strength we have is that backbone. tRNA, we have that backbone, plus we have some hydrogen bonding. rRNA, we're now solid. We're in there with proteins. We like a structure. Okay? Now think about it. Your ribosomes have to be around for a while. Okay? Your ribosomes have a lot of work to do inside your cell. Your tRNA, that needs to be around for a good time because it's got to transfer that amino acid. Okay? It's got to do this whole process of translation quite a lot of times. Messenger RNA is only bringing the code out once. So yeah, he might be used more than once. Okay? We can have polyribosomes and all sorts of fun things going on in translation. But at the end of the day, mRNA is only going to be used to build the proteins while it's needed. So it's not going to last as long. So that's where we're now starting to bring structure and longevity together. mRNA has our least stable structure. It also has our smallest lifespan, for lack of a better word. Okay, so as you increase the longevity of the molecule, you've got to increase its stability. And we can take that all the way to DNA. So if we start from the beginning, we start with our mRNA. Okay, we said this is our weakest and wobbliest one. So we've got no hydrogen bonds, and we've got no proteins. It is our least protected molecule. Then we have our tRNA. Here we have some hydrogen bonds, but we don't have proteins. Ribosomal RNA, here we're making an organelle. Here we have proteins involved. In DNA, we have lots of hydrogen bonds. We have a lot of strength in that double helix protecting that code. We can split this further again, looking at longevity of the cell itself. Prokaryotes, we have no proteins. Eukaryotes, we have proteins. And we have a nucleus. I mean, that's another layer of protection, that nuclear membrane, keeping the genetic material inside the nucleus and further away from what's happening in the cell and from any impact outside the cell. So in a eukaryote, you're really, really protecting that code. And so what we have overall here is an overarching pattern of increasing longevity. Then we need to have increased stability, and therefore reliability. The mRNA that you're using to build proteins at the moment, you might not need that in 20 years' time. But the DNA that's giving the instructions in your cells, in 20 years' time, you're still going to need that exact same DNA with that exact same set of instructions. So it really needs to be much more stable and much more reliable over time. And if we want something to be stable and reliable over time, the structure needs to reflect that. So this is another one of those beautiful points where you're highlighting the fact that structure and function are two things that are really tightly interlinked. You can't separate the two. You've really got to understand structure in terms of function, and you've got to understand function in terms of structure, because the two of them are twined together like Siamese twins. It's always a reason for something. It never happens by accident.